time? Okay, so I'm Deborah Charlesworth, and I would also like to thank the organizers for inviting me to this wonderful place and for letting me listen to all these interesting talks. Now I have to pay it back. So you heard a little bit about animal mating systems, and I'm going to be talking mainly about plant mating systems, but I want to talk about mating systems in a very general way and not specifically about plant ones. So I want to start by reminding you that mating systems evolve in both plants and animals, and just to remind you that hermaphrodite organisms even are not confined to plants or to any particular group of organisms. They crop up in animals, which people rather often forget about. So just to sort of give you the broad view of what we're going to be talking about, this slide summarizes some of the biology. We're really going to get our feet wet with dirty, muddy biology. Some of the crazy things organisms do. I haven't even mentioned asexual organisms, but you've heard about them a fair bit already, and we'll mention them again a little bit. But I'm talking about sexual systems here, and among even sexual systems, there's a good deal of diversity, a good deal of horrible terminology that shouldn't let you, shouldn't disturb you too much, but I have to mention some of the terms. It's convenient to divide things into sexually monomorphic and sexually polymorphic. So starting with the monomorphic organisms, think of a hermaphrodite, like some of the things I showed you. Everybody has male and female functions. As you're aware, many plants would be hermaphrodite like that, but some animals, as I just mentioned. And some hermaphrodites may undergo inbreeding. So the last column in the table there refers to whether inbreeding is possible, not whether it happens. There's also the possibility that you have male and female structures on the same plant. If you're a plant, you can have male flowers separate from female flowers. Let's worry about that. The organism, the individual, is a hermaphrodite, but I may mention this term, monoecious, and please forgive me if I do, because it does apply to plants. But those are sexually monomorphic too. Every individual has male function and female function. And then much more familiar, although the term perhaps seems surprising and unfamiliar, is the condition of being sexually polymorphic. Having the familiar one, of course, is like the humans, males and females separate. And some plants also have separate males and females. You may be familiar with papaya plants, for example. They are male and female. And in date palms, another plant example, in agriculture, you want the fruits of those species, and you would like to have your plants all be female and only enough males to fertilize the flowers and give you fruits. You can have other conditions that are sexually polymorphic as well, and I'll be mentioning these later. You could have hermaphrodites. Some individuals be hermaphrodites, some be female, or hermaphrodites, some be male. And these things also exist. But if you have separate males and females, in this right-hand column about inbreeding, you can't, of course, have inbreeding, at least if it's strict maleness and femaleness. So that's some of the range, at least, and not too much of the terminology. Just to give you a feel for what happens, so we can start talking about evolution in a moment, but we need to have an idea of what actually has evolved. This summarizes various collections of data that people have made about the breeding systems of hermaphroditic plants that can inbreed, can self-fertilize, in fact, and self-fertilization rates can be estimated using genetic markers. As Mike Whitlock explained in his talk just before lunch, if you inbreed, individuals inbreed, you get more homozygotes, and that's the basis for being able to use genetic markers to assess how much inbreeding is going on in natural populations, and I won't go into the technical details. All selfing rates, more or less, are possible, although complete selfing seems to be a great rarity. but anywhere between, if you divide them into rough categories of different levels of self-fertilization, from highly outcrossing to highly outcrossing is on the right, I think, to highly inbreeding on the left, you get species that are represented in all different categories, but there seems to be somewhat of an overrepresentation of extreme values, either highly outcrossing or highly inbreeding, and less in between. That's an old study. This is a more modern study, but you see the same sort of slight deficit of in between. And that's these are partly misleading because we're lumping everything together. There are some patterns which are slightly informative. If you think about annual plants, this one goes the other way, the x-axis. Here on the right, we have an excess of high levels of self-fertilization and not so many highly outcrossing. And that makes sense if you think of an annual plant that has one chance at reproduction, and if the season for flowering and reproducing ends, if it hasn't reproduced, it doesn't get another chance. It would make sense that you would be able to self-fertilize, at least potentially. Whereas long-lived woody species, they get another chance another year, and if outcrossing is better, as Mike Whitlock again explained, the basis of inbreeding depression, that it's usually lowers fitness. If you produce progeny by inbreeding, they have lower fitness than if you produce them by outcrossing. It would make sense that if you have a long life, you would perhaps have a tendency to evolve higher outcrossing. So that's a sort of general picture. And we know that mating systems evolve. You saw that diversity. And what's more, it's not just diversity across the whole of living creatures, but we know many, many cases where closely related species have different mating systems. For example, self-incompatibility, which I won't talk about in detail today, but it prevents self-fertilization. I'll come back to that in my third lecture. It breaks down to self-compatibility pretty often. And this is one reason understanding these evolutionary changes is interesting in itself. As Kavita said, I think this should be the motto for the course. We're curious. So we just want to understand why these things happen. But they're also important for other biologists who don't care that much about evolution. For example, systematists make use of variability to say these are different species, these are the same species, this is a highly variable species, this one is not. And it's been noted for a long time. This is a quotation from the great plant naturalist Herbert Baker. Outbreeding form presents a continuously variable complex of characters. The inbreeder is likely to display a circumscribed set, fewer alleles, fewer combinations of the alleles, and an unsuspecting taxonomist might seize gratefully and happily upon the sharply defined form as a species, and the rest would be regarded as unsatisfactory. So you can see that breeding systems will affect how you do your taxonomic inferences and what you make of diversity. So that's one important thing which I'll come back to. Of course, in horticulture, in agriculture, with plants, humans have made selection. They have selected self-fertile species, much, much easier to breed self-fertile species than ones which require cross-pollination between different individuals. So, for instance, as I mentioned, in breeding date palms, where you have separate sex trees, a lot of effort has gone into carrying branches with flowers from male trees and fertilizing flowers on female trees. And a lot of skill and expertise has had to be developed. How much easier things are when you go to something like rice or wheat, where they self-fertilize and you don't have to trouble yourself. Human effort is not involved. So there are all these kinds of considerations for other biologists and for practical people about mating systems as well. And so what I'm going to talk about is the theoretical framework that can help explain some of these things. So I mentioned that the change from outcrossing to self-fertilization is a common evolutionary change, and also species with separate sexes can go back to hermaphroditism. There are many, many examples of this kind of thing. Some are familiar in model organisms like Caenorhabditis elegans is an inbreeder. Individuals are nearly always hermaphrodites, they're not quite always, that can self-fertilize, whereas other related species in the genus Caenorhabditis are dioecious, they have separate sexes. I'm just getting us used to the terminology for those people who aren't familiar with these things. Another model organism, the plant Arabidopsis thaliana that molecular biologists study, is an inbreeder. It self-fertilizes very quickly in nature and in the greenhouse, but its closest related species are self-incompatible and cannot inbreed at all. So these are examples of closely related species that have different mating systems that we want to understand. And these things are associated with phenotypic changes. The evolution of self-fertilization is very often associated with small flowers. This would be an outcrossing species that is trying to attract pollinators to come and take pollen from one flower to outcross in another flower, whereas these self-fertilize in these small flowers. And this is quite a common kind of difference that you see. This is a completely different genus of plants with an outcrossing and a self-fertilizing, closely related pair of species with that flower size difference. And they go along with other phenotypic differences, like whether they have attractants for insects like nice smell, and different, whether they have color or they're white flowers, that sort of thing. I already mentioned the genus Caenorhabditis, and I'm just showing this slide to show that not only do we know that related species can have different mating systems, but we also know sometimes which direction the changes have been. I just told you earlier that the change from outcrossing to inbreeding is very common. We know that because now we can construct phylogenetic trees, like this one for the genus Caenorhabditis, and we can see that the inbreeders, Caenorhabditis elegans here, which is a hermaphrodite species, and this other one, Caenorhabditis briggsii, those look as if they have been derived from ancestors which were, for most of the other ones that have extant descendants to this day, so the ancestral condition is indicated as being dioecious, having separate sexes. So there are many other examples like this where a phylogenetic tree will tell us that which direction the change has been. It's not just plants, as I mentioned. I will be talking a lot about plants, and that's because a lot of these things have been studied in plants, but it occurs, these same sort of evolutionary changes occur in fungi and are also very important. Here's another example of a phylogenetic tree where it can be inferred where the species has lost incompatibility and has become what the fungal people call homophalic. And you can see several different independent losses in this one genus. That's quite a common sort of observation, frequent breakdown. So before I go to that, I want to just consider these matters more generally. So I'm going to need bigger piece of chalk than that. Right. So we have asexual versus not asexual versus sexual. We have, well, I should say inbreeding, self-fertilization. These might be hermaphrodite, well, these would not be dioecious. Versus outcrossing, and we have dioecious. Can't write on the blackboard very well, which would mean male and female, separate individuals, right? And now let's think about the factors that will give these different situations advantages. So I want to think about, for example, considering an individual that's asexual and an individual, say, of the same species that's a hermaphrodite that can reproduce sexually. What would be good about being asexual compared with reproducing sexually. What factors might have influenced which would be better if you were just to compare two individuals you're looking at now? Sorry, you have to speak louder because from here I can hear see why the other speakers didn't catch the questions. There's a loud fan. Yes, so mate availability. Asexuals, it's very good. They have good mate availability. They just produce an offspring all by themselves, same genotype as themselves, for example. Self-fertilization, how is mate availability for a self-fertilizing organism? Good, bad? It's usually good. It's a bit less clear because self-fertilization might sometimes involve having to have an insect visit and push pollen, say, onto the stigma. I will talk about plant examples and cases. Oh, now we'll have quiet and we'll get too hot. Anyway, how about separate sexes? Mate availability good? It depends, yeah, but it could be bad. And so it's a factor. So this would be good, I should say, really. This would be okay to good. This might be sometimes bad. It might depend on, for example, the density of the pollinating insect, if it needs an insect. Or it might depend on the density of conspecific individuals, right? If you're a female and there's no males, it's bad. So mate availability, that's an important factor. What else? Sorry, I think I heard it. Say it loud. Sorry? Immunity. I'm not sure what immunity we're talking about here. No, that one doesn't correspond with my little list I have in my head anyway. Remember Mike Whitlock's talk? And I just said some of them are good at inbreeding and some of... Now I'm just looking at one individual and of each alternative type. Let's worry about inbreeding too much. It's true that, you know, if you self-fertilize, if you are a homozygote, your progeny could have the same genotype as yourself. But it's a factor I'll come back to, but we needn't worry too much about it. If we're just looking at the progeny of one mode of reproduction right now. Well, Mike Whitlock was talking about inbreeding depression. So is inbreeding depression a problem for asexuals? So I should talk about progeny survival. What about asexuals? What might affect their progeny survival? It just depends on the genotype of the parent. It'll be the same. But is there inbreeding? Who thinks there'll be inbreeding when an asexual reproduces? Yeah, but I'm not talking about the long term. I'm talking about just two individuals now, and we're looking at their breeding and producing their offspring. So it's just, you know, it's the same as parents. Should be okay. What about self-fertilization? Yeah, so there's inbreeding depression. Perhaps. And what about outcrossing? Yeah, no inbreeding depression. So those are important factors. This is an ecological factor if we want to try and subdivide things according to the different sort of categories. And this is a genetic factor. Yeah? Anything else? There are two more that I... Yeah, I'm just being crude here. I'll come to that. I'm just being crude here. I'm saying self-fertilization, it could be high level, low level, but just the potential to do some self-fertilization. It can give you better mate availability, which we call reproductive assurance, compared with not being able to do any self-fertilization because you have separate sexes or because you're self-incompatible, which I'll come back to in lecture 3. But these are just some crude possibilities. Well, there are two more that were not considered here so far. One of them is the allocation of resources. To reproduction. And I showed you some examples of self-fertilizing organisms. And you could see from those examples, I pointed out that the flowers are often small and they often don't produce scent or nectar to attract pollinating insects, for example. So they can actually, self-fertilizing organisms can perhaps have small flowers. So if you have a small flowered mutant, it could self-fertilize with lower resources than an outcrosser. So that would be a factor that might come in. It could be economical in terms of resources compared with outcrossing. That's one factor. And similarly, when we think of male and female, the resources available to the individual at the time of reproduction might differ. So there might be some effect, which we needn't specify now. I'll come to that in the second lecture. I'll talk about it a bit later today. But so these are some of the factors, right? These are all sexually reproducing. Let's be. Yeah. No, not the economic argument as far as males and females. You may be able to produce more flowers, for example, if you're a male, because you don't have to nourish those flowers and give them resources as the seeds develop and the fruits develop. Think of something with large fruits. Perhaps a male can reproduce well with a lower investment, but a female would need a big investment or big amount of resources to draw on. So you could have those factors. So just these factors alone tell us something interesting, which is that what I just wrote here, that we can make a division between sexual and asexual, which is obvious and has been done many times during this course already, but self-fertilization is not the same as asexual reproduction. It is influenced by quite different factors. And if you think to be completely concrete, an asexual genotype is, for example, in real biology, asexual genotypes are often hybrids between two different species, for example, and they're highly heterozygous. They have one genome from one species, one from another, and if they're lucky and are able to reproduce at all, sometimes it's only through the asexual mode of reproduction. And they produce offspring which have the same genotype as themselves, also heterozygous, whereas a self-fertilizing individual will produce offspring that are more, if it reproduces by self-fertilization, it will produce offspring that are more homozygous. So this is a very big difference, increased homozygosity with self-fertilization, but not with asexual reproduction. So although there are some things which are quite similar between the two, there is a fundamental difference, and one should not call self-fertilization asexual reproduction. It's a form of respectable sexual reproduction, but the organism is just mating its own male and female gametes together. That's an important distinction. Okay, so these are not, in fact, all the factors. If you listened attentively to Professor Joshi's wonderful lecture the other day, where he categorized all the different factors that you could think of in one generation, affecting the transmission of genes from one generation to another, I think he was pretty careful to talk about random mating. And there's another factor which hasn't been considered here, which is the actual transmission of genes, which in random mating is going to be the same for the different sexual systems here. But when you think about asexual reproduction or self-fertilization, then there comes in an important, interesting difference, and that's where we get back to these. You can think, first of all, it's simplest to think about asexual reproduction, where the individual reproduces its own genotype. The two genes that every progeny individual, the two alleles at a locus that every progeny individual receives, both come from the same parent. And it was pointed out a good while ago that if an individual, well, you can regard really outcrossing as sharing your representation of genes in the next generation with someone else. Asexuals are in a way selfish. They don't share that. They contribute two genes from their own genotype, from their own genome, and so they're selfish in that respect. And if you think about it, you can see that an individual, if it, instead of contributing one gene to an offspring, sharing the other, say, is from a male that fertilizes it, if instead it reproduces as a female, produces asexually offspring with two genes, then the frequency of its genes has doubled in one generation. And you can do the little bit of population genetics for that case, and it's in the textbook that you don't have copies of, but there is a copy floating around. In our chapter 9, we have the very, very simple little bit of population genetics to show that result. And of course, an asexual therefore has a big advantage. But the case I want to show you is slightly more juicy in terms of population genetics, and that's the model for the evolution of self-fertilization. And this was published a long time ago too, in 1941, in a paper by R.A. Fisher. So Professor Joshi's talk was all sorts of wonderful things that Fisher did. So don't get away with the idea that Fisher didn't know about this one too. He had this idea too. He published in a paper which wouldn't lead you to expect, that's the title of the paper there, Average Excess and Average Effect for Gene Substitution. You wouldn't have thought that it would deal with the evolution of mating systems. And for a very long time, this paper was a very important paper in quantitative genetics and was cited by people who worked on quantitative genetics. And then in the 1980s, people started or people remembered that it had been, had an aspect to do with mating system evolution, and the two are very closely connected, in fact. So again, to give you an intuitive statement of what's going on, it's very much like the one I just told you for asexual reproduction. Consider a population of self-incompatible hermaphrodites in a constant population of such creatures, and an individual contributes two genomes to the next generation, one as a male and one as a female, outcrossing population. If you had a mutant which is capable of self-fertilization, it's going to contribute genomes to the self-fertilized progeny. Both the genomes present in the self-fertilized progeny come from that individual, and it could still, at least in a theoretical model, and sometimes in real life, it could also outcross the gametes of another individual through its male gametes, and so it could contribute a total of 50% more gametes to the next generation than the individuals in the starting population who only outcross. That's a simple way to say what's happening. I'd hoped there would be more Blackboard and I could leave this up, because I bet I need to come back to this. But at any rate, what I should have said is, so we could add a fourth column here, transmission of genes to the next generation. Right, so let's look at a model. So we have genotype, just the usual sort of, oh dear, population genetics. A1, 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 A2. Like that. Right? And we need to have the rate of self-fertilization, which we call the selfing rate, to save time. And we can imagine that this is the original outcrossing population, so it has a selfing rate of 0, and we can imagine that this mutant produces a phenotype that completely self-fertilizes, like in that simple word model. And we start with the frequencies of the genotypes, and we call them X, Y, and Z. This is just following exactly what's in a box in our textbook. And so what we're assuming here is that all of these different phenotypes produce exactly the same amount of eggs, of progeny as females, and it makes no difference to the amount of pollen that they contribute that will fertilize of other individuals, and provided the available eggs of other individuals. So this is a model that's of mating, very crude, simple model, that's called the pollen pool model, where all of the individuals that can produce any male gametes or pollen, to use the plant terminology, it goes into a pool, and that gets carried by the wind, by pollinators, and a well-mixed pool is assumed, which of course is nonsense in biological reality, and it gets, in proportion to the allele frequencies, onto the recipient individuals to fertilize their female gametes. And so what we could do is we can write down the frequencies in the next generation, thinking about the genotypes that are produced by each individual, each genotype. So this is the frequency in parents. And then we can go to the frequency in the progeny generation, the next generation. So, for example, that will come about this genotype in the progeny. Well, how will this genotype be produced in the progeny, A1, A1? Can it be produced by that parent genotype by self-fertilization? No, because it's got a selfing rate of 0. So it can only be produced by this type of genotype through outcrossing. Are you with me? So where will it get? It will get an A1 from its parent of this kind, of the same as that genotype. Where will it get the other A1 allele? Yeah, from the pollen pool, the component of the pollen pool that's contributed by those, exactly, right. And so you can write it down. I haven't got room on the board or time and so on to write it all down, but you can see you have to patiently work them out one at a time. For each progeny genotype, you have to consider the contribution from each parent genotype through outcrossing, say, and through self-fertilization. It's easier to start with self-fertilization, write down the contribution that will come, and then the contribution that will come through outcrossing, assuming this pollen pool model. And if you do all of that, or if you look in the textbook to see it, it's very simple to do it. It's just tedious and takes more blackboard space. What you come out with is that the frequency, so I didn't write down the frequency of the A1 and A2 alleles. We're going to call it, I think we're going to call it Q, A2 allele, sorry. And what we come out with is that the frequency in the next generation is, well, approximately like that. And that's just what we said before, that for every 2 copies transmitted by an outcrossing individual to the next generation. This selfing one contributes 3. This is an extremely crude model. This is assuming that it's at very low frequency at the beginning. But it always increases in frequency. It would spread through the population under this crude model. Of course, you could easily extend the model to say this isn't completely outcrossing, this doesn't cause complete self-fertilization, and all those things have been thought about. And you can have various refinements of the model. But the important thing is to see that there can be a very large transmission advantage, similar to what I said for the asexual case, from one generation to the next, a very large transmission advantage to modes of reproduction that contribute more copies from the one parent type than another. And this was not included in the various kinds of selection stages that we heard about up till now, I think. We heard about survival from the zygote stage, from juvenile, say, to adult. We heard about fertility differences, but here in this model, as I said, we've made the assumption that there aren't any actual differences in fertility. Everybody's equally fertile as male and female, but the mode of transmission is causing this advantage. So that's a new thing, and it sort of ties in again with the lecture we had about think about your assumptions. You might think you've covered everything, and then somebody clever like Fisher comes along and realizes that there's something else that hasn't been covered. So I think that's that one. This was done now. Maybe I mentioned, I called it Fisher's model. A refinement that is very sort of biologically obvious was added by Tom Nagalaki. So even these very simple models actually have not been beneath the attention of pretty sophisticated mathematical people. And it's funny that the more interesting biological insights, these are very important biological insights, have come from the mathematicians. What Tom Nagalaki added to this is the important point that I've already mentioned as an assumption, that each of these genotypes is supposed to do as well in both male and female reproduction as the original one. Well, they're all supposed to do as well. But of course, in reality, if pollen in a plant, for example, is used for self-fertilization, that might well have the consequence that there's less pollen available to go into the pollen pool. And Nagalaki added that important concept, which is called pollen discounting. So this approach helps us understand asexual reproduction, this transmission advantage, and self-fertilization. And it shows us that there's potentially very strong advantage to either of those modes of reproduction, and yet asexual reproduction is far from prevalent, and even self-fertilization is not the norm, as you saw in those empirical data. Another thing that the model makes clear is that male reproduction has to be taken into account when we model the mating system. And it gives us the insight that if you want to call this sort of thing reproductive fitness, so as an extra sort of fitness that we didn't hear about in the earlier models, we can't forget about male reproduction. I don't think that anybody theoretical would ever have written down a model that forgot about male reproduction, but one has to admit that the botanists for a long time were inclined to call seed output or female egg production fitness. And that should be a sort of interesting little side issue that one should be aware of and be careful about not ignoring important things that would contribute to fitness. And the fact that there's this transmission advantage that, as I said, could be a large selective advantage to a changed reproductive phenotype is, of course, relevant to the fact that I already showed you that changes from outcrossing to self-fertilization occur very frequently. it makes sense of that observation. And even more fundamentally and importantly, this understanding of transmission advantage led to a real change in the entire field of understanding mating system evolution, because it's looking at the thing in a simple, one generation to the next generation, individual selection perspective. So I've already said some of these assumptions, and as I said, the models can be extended. But these general conclusions from the models that I just highlighted are not affected by the exact details that you come to. I just show you for fun that this is what it looks like when Fisher worked it out. And in our book, it's not that complicated. I'll come to some simplifications even over what we have in our book, or over the one I showed you, over this population genetics. I've already mentioned this pollen discounting. If you evolve self-fertilization in such a way that you use all your pollen in a plant, for example, in self-fertilization, then of course that's going to make a smaller advantage than what I showed you. And that's quite possible. There are plants, oh dear, plants that have this horrible terminology, cleistogamous flowers. And all it means is that the plant self-fertilizes in the bud. The bud doesn't really open. The pollen is dumped on the stigma of the plant and it does the fertilization. No pollen is exported into the pollen pool or onto the stigmas of other individuals. So it's quite realistic to believe that sometimes evolving, or a mutant that can self-fertilize, might, right from the outset, do it through some phenotypic change, like not opening the flowers, that cause this to be complete pollen discounting. In Caenorhabditis elegans, hermaphrodite, it's similar. The individuals can self-fertilize. They're hermaphroditic, they can self-fertilize, but a hermaphrodite individual can't use its male gametes to fertilize a different hermaphrodite individual in that species, and that obviously is going to make a big difference and really reduce the advantage of self-fertilization. On the other hand, still the ecological advantages of reproductive assurance, of not needing to find a mate in low density, those things would still be advantages for self-fertilization. In the context of the animal mating systems that Dr. Vidia talked about the other day, we can have this same sort of phenomenon. I'm not going to talk much about animals because these sort of mating system changes are so much more studied in plants, and in a way more dramatic. Self-fertilization is much more dramatic than mating with a cousin or a sibling even, and easier therefore to detect the biological effects. In an animal, mating with a relative, though, would often use up a mating opportunity that could have been mating with a non-relative. And so this discounting will still will come in these animal cases too. So why are we not all self-fertilizing hermaphrodites, given this big advantage which is often present? And the obvious question, the obvious answer to that question might well be that inbreeding depression, another genetic factor, opposes the transmission advantage. So we have two opposing genetic factors, and whichever one wins, you could go one way or the other. And we know that inbreeding depression is very important. Mike Whitlock already talked about it for animal and plant breeders. I think he probably really mentioned it in conservation biology, as well as the evolution of breeding systems. So I'm going to go quickly through it. The one on the left is a picture of real maize individuals starting with two inbred parents, and F1 showing hybrid vigor, and then subsequent generations vigor and size and everything going down. The one on the right isn't the same picture Mike showed, which was the inbreeding coefficient gets bigger on the x-axis, and the quality of the progeny goes down. This is a different experiment entirely, but it looks exactly the same. This is the seed germination rate. So even at that early stage of the life cycle, we're seeing inbreeding depression with low quality seeds that germinate at a low frequency when you have the parents mated such that the offspring will have a high inbreeding coefficient. It's not just in that in those species. This is a study in the field by John Willis showing inbreeding depression not just at the germination stage where it's prominent in the experiment I just showed you, but not so important in this particular case, but survival. These are two different populations, the two different symbols, but it's occurring in both populations. And as you go through the life cycle, you see more and more effects of inbreeding at every stage. And I just show a picture of lethal mutants when you do self-fertilization. Of course, if the parent is heterozygous for a lethal mutant allele, you'll get a quarter of the offspring being homozygotes. And sometimes you do see that these are white seedlings that have no future. They can live to this stage on the reserves in the seeds, but then they're going to die because they can't photosynthesize. So all sorts of different sizes of mutational effects and all sorts of different life history characters are showing inbreeding depression in experiments. And again, as Mike mentioned it briefly, we expect the inbreeding depression, it's due to these different kinds of genetic factors that I just mentioned, and it will change as the mating system evolves. In reality, this just shows some evidence that it does change. Outcrossing rates for different species in this Hawaiian genus of plant, Schedia. This is from the work of Steve Weller and Anne Sakai, who studied mating system evolution in this genus. You go from highly selfing to mostly outcrossing, and you can see more inbreeding depression. These are various different measures of inbreeding depression at different times, different characters, different stages of the life cycle. They tend to be higher. You can see a clear tendency for them to be higher in the highly selfing species compared with the more outcrossing species. And that's what you would expect, just as Mike explained, with homozygous recessive or fairly recessive mutations that are present in the population will be removed by natural selection, seeing them in an inbreeding population much more than in an outcrossing population. So I maybe should stop at that point and make sure nothing I need to explain that's totally obscure to people. Are there any questions about this idea? Okay, it's very simple stuff. So we did this population genetics, or I hope you will do it, or at least look it up in the textbook, but right through instead of just the outline of what is done. A person called David Lloyd came along in the 1970s and thought up a clever way to bypass the complexities of the genetic models of self-fertilization by using phenotypic models of natural selection. And I want to explain that briefly to you because I've already given you the concept of reproductive fitness. And what David realized is that you can define fitnesses of phenotypes with different reproductive behaviors, and you can use those to analyze these models, bypassing a lot of the complexity. And the idea is that fitness through reproduction can be defined as the number of gametes contributed to the next generation, as we've really been seeing in the population genetics model. And so if two phenotypes have the same fitness, provided the assumptions underlying this are okay, there would be no evolutionary change. But if one phenotype had higher fitness, then it would increase in frequency. And obviously the genetic factors that cause that would increase in frequency. And using this simplification, which we'll study, we'll look at a bit more carefully in the tutorial. I won't go into it now. Lloyd was able to then deal with more biologically realistic models for self-fertilization. What we had was very crude. You may have thought it was fine, and I gave you some extensions to it, but actually when you think about it, it was very crude indeed. We just assumed that each genotype has a self-fertilization rate. And Lloyd realized that self-fertilization in the real world, and now we're really talking about plants, but similar concepts would apply in an animal, could at least be divided into different categories. competing self-fertilization, for example, where pollen is mixed by, say, a pollinator visit. Pollen of the individual itself gets mixed with the pollen pool that's coming in, and you would have some kind of a mass action situation. Or you could have, of course, prior self-fertilization, where each genotype just has a self-fertilization rate, simple model. Or you could have delayed selfing, where the plants are clever and they organize their flower structure, for example, in such a way that self-fertilization is avoided. You notice in many plants that they keep their pollen well away from their stigmatic surfaces, but sometimes they wait and they wait, and a pollinator doesn't bring pollen to them, or when they don't receive pollen from outside, and then the flower has a developmental process that will bring the anthers close and dump pollen on the stigma. So that would be delayed self-fertilization. And the way Lloyd's models work, I gave a reference in the readings to his paper, which is not entirely easy to read because he was the first person really introducing these ways of doing it, and so it's kind of crude and clumsy, some of the ways he does it, but it's simple-minded. You just have to plod through it. But he assumed that there are two phenotypes. So, for example, if we have the three genotypes here, we had two phenotypes. We assumed that A2 was dominant. And we can calculate their fitnesses, and we can assume they have some number of ovules that get fertilized. We can assume that so the other two phenotypes, they have some proportion of ovules that will be self-fertilized, either within a flower. All sorts of different processes could go on, and they might all go on in one species, but we could consider parameters for each of the possibilities. The prior selfing, the ovules will be self-fertilized before pollen arrives in one phenotype, but maybe not in another phenotype. And we're going to compare the fitnesses. We also need to know the proportion of ovules that are fertilized by another flower, and we can get the reproductive fitnesses. And just to give you one example, Lloyd's competing self-fertilization model, these are what you get from those parameters. It looks a mess, but it's very, very simple, of course. And then you can look at the difference in fitness through reproductive functions, and you get something out at the other end. And these are the results that I've summarized from Lloyd's paper. And what Lloyd did with this is extremely interesting and important. He compared the fitnesses, like I've just shown you, simplifying the population genetics by assuming just two phenotypes, but otherwise showing that the phenotypic fitness approach is correct. And in fact, in one of his papers, Brian provided an appendix showing that it gives identical results under those assumptions to what you would get if you work out the full population genetics without evading those complexities. And so you get these, it doesn't matter what the details of them are, but you can get expressions for the fitness differences. And what Lloyd did was he compared that with what you would get if you compared two populations, one with one mode of reproduction and one with the other. And they're not the same. In competing self-fertilization, you get self-fertilization is eliminated if this the fitness of the inbred is greater than 0, whereas you get a different thing here. With prior self-fertilization, you get a different, in delayed self-fertilization, whether you look at the population, fitness of a population that all have one phenotype versus the other, it comes out the same as the individual selection model. Selfing is always advantageous. That's not surprising because if you have delayed self-fertilization means that if you haven't got any reproductive success at all without self-fertilizing, some self-fertilization would be good even if there's inbreeding depression and the survival of inbreds, which is I, is less than, is low. So Lloyd this paper was a real landmark paper because it showed that you get a different result if you compare the mean fitnesses of populations with what you get if you actually work out the population genetics. And that's because the population genetics is considering this transmission difference that we've seen is important. So I'll come back to that in a moment and later. I've said that we ignored some important biology. Pollen discounting doesn't make a very big difference. You can also assume something to do with the amount of selfing and how much pollen is left, how strong discounting is, how much pollen is left to go in the pollen pool. And when you do that, you can actually, under this competing self-fertilization, it turns out you can get a polymorphism with an intermediate level, or you can get an intermediate level, I should say. It could be a polymorphism for inbreeders and outbreeders, or it could be an intermediate level of self-fertilization that's exhibited by the population. Those are important insights, given that we see that intermediate selfing rates empirically are common. They're not inexplicable. Diversity is, I've already mentioned increased isolation, is that you have homozygous genotypes, so you don't have alleles at different loci recombining. If individuals are isolated in different separate homozygous genotypes, you don't have double heterozygotes at two loci, even though you may have, within the species or population, you may have genetic variants at two loci. You just transmit homozygous genotypes to the extent that outcrossing doesn't occur. And so any processes that require recombination are altered in inbreeding populations. For example, when an advantageous mutation spreads, selective sweep occurs, and you could get changes in allele frequency through that sort of process, and you'll hear more about that during the course. But they become, in some ways, more like an asexual population, and some of the things that Lindy has been talking about would be seen. So recombination is reduced in inbreeding populations, and that will also reduced will also lower diversity. Variants are likely to be removed by selective effects. You'll hear more about that. And so with reduced diversity within populations and increased isolation between populations, those two things come together to increase subdivision. In measurable terms, and again Mike will explain how you can measure the degree of subdivision between populations using genetic variants, but we will see that. In addition, population bottlenecks, which are just mentioned somewhere here, I think, a self-fertilizing population can found a new self-fertilizing genotype can found a new population through a single individual, and you could have a real population bottleneck adding to the lower NE, because the real population size is small, the NE is small. So all sorts of things will go on once a change in the mating system to a higher degree of inbreeding occurs, or obviously the other way around. And there's just some empirical data showing you that this happens. This is a set of data from outcrossing plants here on the left-hand side. These are data from different genera, and these are showing the within species, with the total within species diversity, allozyme diversity, and the within population diversity. You can see that they're very much the same. Outcrossing plants, you find within a population much of the diversity you would find in the species as a whole, and this is just showing you the ratio of the two. Whereas in inbreeding plants, you often have species, well, there are lots. If you look at the mean here on the right, the diversity within populations is often a lot lower than if you take the species as a whole and estimate the diversity taking samples from multiple populations. And that's just showing you the ratio of the within population to the total. It's not only with allozymes. You see exactly the same kind of phenomenon with DNA sequence diversity. This is in some tomato species growing in South America. And this is the within population diversity. The outcrossing species have high diversity over here. These are just estimates for various different loci, sequencing them and estimating diversity. And the inbreeding species over here, which is just the species name, they have very low diversity. So it's exactly the same phenomenon. Low diversity within populations or species when you have high inbreeding levels. And this is just another example to show you it's not just a plant thing. This is Caenorhabditis species. Here's an outcrossing species, and this is C. elegans, this inbreeding species. This is within population diversity. And here we have the contrast, a measure of population subdivision based on, again, a set of loci. The outcrossing species have low subdivision. The inbreeding species show extremely strong evidence of subdivision. So I've really said all this already. This is my conclusion. We can see these consequences in the long term, but the advantages also of the race are long-term consequences, I think, and Fisher understood much more clearly than other evolutionary biologists that the individual selection advantage is what you have to look at if you want to see what evolutionary change happens. And Fisher, I highlight this quotation from Fisher, the principle of natural selection refers only to the variation among individuals or cooperative communities and to the progressive modification of structure or function only insofar as these are of advantage to the individual. David Lloyd, again, same quotation, I said before, is emphasizing that now we have a really clear example. In fact, it was Fisher's original intention, his original example. One of the things he's doing in that paper was showing the importance of the individual selection advantage, even though we see long-term consequences, so we can do correlates and see things. But those things are not, that are correlated, are not necessarily, in this case, not why the change evolved in the first place. So I think I've got left some time for questions, if there are any. We just have to have some questions to fill the time. Or comments? I have a horrible feeling Brian wants to make a comment again. And just do it by making small flowers and be successful. And then there are other ecological settings in which selfing could be, there could be what one might consider an evolutionary filter, where only selfing organisms can establish, and that would be situations where reproductive assurance is important. For example, Herbert Baker, who I've mentioned, one of the things that he noticed was if you look at islands, plant communities, you see self-fertilization is rather commoner than you're used to. And he realized that species, or even genotypes, members of species that have the ability, be mutants or whatever, to self-fertilize, those will be able to establish a new population, whereas some genotype that's absolutely required to outcrop would be unable to do so. So reproductive assurance, reproduction on your own, reproduction in the absence of a good pollinator service, reproduction under colonizing conditions also can, these sorts of situations can arise. These ecological conditions can make the advantages for selfing, can add to the advantages of selfing on top of the transmission advantage. That makes it even more clear that there are individual selection advantages, and they can outweigh inbreeding depression so long as some progeny can be produced. Is that enough, do you think, to I had mentioned those things earlier, but it's, I was going to actually sort of synthesize this at the end and say that another thing I haven't mentioned here, but you might if you were listening carefully and not having a little nap here, that when we're thinking about these allocation problems, we're thinking about a situation where selection is frequency dependent. What you do depends on others. How much pollen you should produce if you're trading off and you're looking at an allocation strategy between female and male functions, what you will do depends on what others are doing. If there are plenty of available non-self-fertilized ovules, it would be a good strategy to produce more male gametes. But if the others are self-fertilizing, then it's a good strategy to reduce and concentrate on female fertility. So I didn't explicitly mention frequency-dependent selection, but it has been mentioned a few times, and it does implicitly come in here, and come in more when I talk about other mating systems. So I mean here, for example, I haven't talked about how self-fertilization versus outcrossing is done. I said maybe you can, a mutant could be a self-fertilizer because it makes small flowers and its pollen is produced so close to its stigma that that's what it does. That is one possibility, but that's the only biological detail I've given you about how an organism might do this. And we'll come to other much more biologically realistic situations and the few left. I don't know if anybody else has comments or